Hello, I am Awantika Mishra from Dr. Hari Singh Gaur University, Sagar, studying BSc 5th Sem CFZ. I am here to present my zoology mid-second assignment under the supervision of our respected Dipali Chat ma'am from Department of Zoology. My topic is antibody-dependent cell-mediated cytotoxicity. These are the contents of my topic. Before describing the antibody-dependent cell-mediated cytotoxicity, I am going to define cytotoxicity. Cytotoxicity is the quality of being toxic to cells. Under the cytotoxicity, there are two terms, necrosis and apoptosis, which I am going to define. The first one, necrosis. Necrosis is where cell lose membrane integrity and die rapidly as cell lysis. Second one is apoptosis. Apoptosis is programmed cell death, where the cytological and molecular event occur inside the cell. They will shut down the metabolism, lose membrane integrity and lyse. Now come to our main topic, antibody dependent cell mediated cytotoxicity. ADCC. What is ADCC? ADCC is where the effector cell lyses the target cell, in which membrane surface antigen bound to the antibody, which is actually the part of humoral immune response. In the next part, I am going to describe some terms which help us in understanding the mechanism of ADCC, antibody dependent cell mediated cytotoxicity. So the first one is MHC, major histocompatibility complex. Major histocompatibility complex is a genetic complex with multiple loci. In simple words, it is a group of genes found on the surface of cell of the immune system to recognize the foreign substance. It is present in higher vertebrate. In human, it is called as human leukocyte antigen system or HLA system. Now there are two classes of MHC, class 1 and class 2. Our major concern for ADCC is class 2. Now, what kind of cells present in class 2 of MHC? These are professional antigen presenting cells. Now the second question arises, what are these antigen presenting cells, or which cells are the antigen presenting cells? These are dendritic cells, mononuclear phagocytes, some endothelial cells, thymic epithelial cells and B cells. These cells help in initiating the immune response. Now let us talk about the structure of MHC class 2. This is the structure of MHC class 2. It is a heterodimer with alpha and beta peptide chains, but it is also have homogeneous like alpha 1 and alpha 2, beta 1 and beta 2 peptide chains. Because the antigen binding groove of MHC class 2 molecule is open at both ends, while the corresponding groove on class 1 molecule is closed at each end, the antigen presented by class 2 molecules are longer, generally between 50 and 24 acid residues long. Now the second term is Fc region of antibody. This is the structure of antibody. This half blue and half red chain is the light chain, and the red region of this chain is known as the variable region. The whole upper region is known as the Fab region. The lower region, the whole blue part chain is the heavy chain, and this region is our major concerning region, that is Fc region. What the term Fc means? Fc means is fragment crystallizable region. It is composed of two heavy chains that contribute 2 or 3 constant domains depending on the class of the antibody. This upper part Fab, and the lower one is Fc. By binding to specific protein, the Fc region ensures that each antibody generates an appropriate immune response for a given antigen. Now let's come to our main topic after knowing these terms, that is antibody dependent cell mediated cytotoxicity. There are some cells in the immune systems which help in antibody dependent cell mediated cytotoxicity. What are these cells? These cells are natural killer cells, macrophages, monocytes, neutrophils and eosinophils. Now let us see how these cells helps in antibody dependent cell mediated cytotoxicity. NK cells, natural killer cell, expresses the Fc gamma receptor. Actually, the Fc receptor in the NK cell is CD16 or Fc gamma R third, which recognize the Fc receptor of the antibody and release cytotoxic factors which cause the death of the target cell. When virus infect the host cell, at the time of replication, the viral protein expressed on the cell surface membrane so that the antibody bind to these antigen. In the next step, NK cell come and the receptor Fc receptor of the NK cell recognize the antibody, binds to it, and NK cell release the cytotoxic factor, that is perforin and Gren enzyme, which cause the lysis of the cell and hinder the spread of the virus. We can simplify it through the diagram. This orange color is the NK cell, this grey color is the tumor cell. This is the Gren enzyme perforin, that is the cytotoxic factor present inside the NK cells. This is the antibody which is recognized by the Fc receptor present in the NK cell, that is CD16. The perforin remain in the cell membrane. The Gren enzyme goes inside the cell and cause the lysis of this tumor cell. Our next cell is eosinophils. There are some large parasites like helminthes that are too big to be engulfed and killed by phagocytosis, and in addition, its outer integument is resistant to the attack by the substance or cytotoxic substance released by neutrophils and macrophages. There is a IgE coat present in these parasites. The Fc receptor of the eosinophil is Fc epsilon R1, which recognizes the IgE coat of the parasite, and the interaction between this cause the lysis of cell. See in this diagram. This is the effector cell. The purple colors are the cytotoxic granules. These are the free antibodies. These black colors, solid black colors, present the Fc receptor in the effector cells. These brown dots are the antigen present on the cell membrane. This antibody binds to these antigens, and this Fc region recognized by the Fc receptor present on the effector cells. This whole process leads to the antibody dependent cell mediated cytotoxicity. Now let's come to the another slide, that is in vitro assay. When macrophages, neutrophil, eosinophil bind to the target cell, the metabolism of the cell increases, or we can say they become more metabolically active. So as a result, lytic enzyme in their cytoplasmic lysosomes or granules increases. Activated monocytes, macrophages and natural killer cells secrete tumor necrosis factors. They cause the perforin mediated cytotoxicity similar to CTL, cytotoxic T lymphocyte mediated cytotoxicity. Here we will understand it through the diagram. See, this is the purple color antibody, green color tumor cell. These antibody come bind to the cell surface antigen. Then NK cell, Fc receptor is CD16, come this orange color. Fc receptor recognize the purple color antibody, bind to it. This cross linkage of CD16 triggers degranulation into a lytic synapse. Tumor cells die by apoptosis. In the next slide, we will see the medical applications of ADCC. Natural killer cells involved in killing tumor. Natural killer cells used in ADCC as a less discriminate cell destroyer than antibody specific apoptosis mechanism. ADCC helps in regulating metastatic tumor using interleukin protein to activate the cell. This is the diagram which also help in clarify this mechanism. This is the antibody, black color, in purple color tumor cell. Dots that present on the cell surface are the antigen. The solid black color antibody binds to this antigen, then this green color, light green color NK cell come. These are the CD16 Fc receptor recognize antibody bind to the antigen. This cross linkage occur and cell dies by the apoptosis. Now at last I would like to tell some of the significance of ADCC. Due to the IgG antibody, ADCC antibody dependent cell mediated cytotoxicity is more useful in viral infection than bacterial. Antibody dependent cell mediated cytotoxicity helps in removing outside toxins. Antibody dependent cell mediated cytotoxicity helps in the formation of antibody, which leads to the use of vaccine and killing antigens. Thank you so much.